Thanks, everybody, for attending today, especially before the holiday break. It's my pleasure to introduce Dr. Jack Foster, who is presenting here today. He received his bachelor's in biology from Northwestern University. He has his PhD in ecology and evolutionary biology from the University of Illinois. He's a professor in the Department of Biological Sciences at Northern Arizona University, where he is the head of the FOS lab. His work focuses on pathogen evolution and disease ecology in both wildlife and livestock, and he's currently on sabbatical working here in Hawaii on projects involving seed dispersal, avian malaria, and bird and rodent diets. So thank you, Dr. Foster, for joining us today, and we're super happy to have you.
All right, thank you.
Thank you for coming. This is a leg.
All right.
Token of our appreciation.
Yes.
And you also get one more
Yeah.
because you're so special.
All right, thank you very much. All right, I can find everything on my desktop except for my presentation. Oh, this is kind of funny. All right, oh, I don't know what I did. Can you see it? No, maybe I need to share it.
Not yet, you need to share a screen.
Okay.
Recording in progress.
And if we mute any other computers that have it going in the room, just to make sure that we don't get an echo.
Okay, can you see it?
I
Thanks.
can see it.
Okay, perfect. All right. It all works. Thank you. Thank you so much for the invitation. It's great to be here presenting some of our research. And so, yeah, scatological adventures to understand food webs. And if you don't know what scatological means, you'll find out in a second. And so I have a bit of a kind of intro to myself. And so mostly to the students here to kind of see my career trajectory. I actually started before many of you were born at Hakalau, where I was working at a project from USGS looking at how rat predation is affecting native Hawaiian birds. And so after that, I worked at a bunch of different sites, including in Alaska. And then I had a position in Saipan, where I was working on the endangered nightingale reed warbler. I also worked for a couple of years on Kauai, on the small Kauai thrush, or the puaiohi. And I also did an introduced bird database analysis. And so I've worked on 5 of the main Hawaiian Islands. So I've had kind of great fortune to be able to work in lots of different places. And so, and most recently, or after that, I had a postdoc at the Smithsonian looking at avian malaria, and this amazing picture of Culex mosquito feeding on the eye of a apapane from Jack Jeffrey. And I continued to do that avian malaria work. And yeah, so when I was thinking about graduate school, you know, I had been 5 years between undergrad and grad school trying to figure out exactly what I wanted to do. I wanted to work on Hawaiian birds, and my advisor was like, no way, in terms of what I wanted to work on. Just the sample sizes were really, really small. Even then, this was 25 years ago. And so sample sizes were incredibly small at the time. They're even smaller now. And so I was pushed to think more broadly and to look at introduced birds. And so over 178 bird species have been introduced to Hawaii. I think it's 180 something now. 55 of those species have become naturalized and so have breeding populations here now. And so there's lots of introduced birds to be able to look at. And so I had an initial idea. I wanted, and this is actually what funded my entire PhD. I wanted to look at the invasion of Japanese bush warblers across the island. So they were introduced from Japan in 1929, and they stayed on the island of Oahu for almost 5 decades, and then subsequently spread to Molokai and then Maui and the rest of Maui Nui in the late 70s, early 80s. They spread to the Big Island in 1997, and they spread to Kauai in 1983. But even though it funded it, I really could not do that project. It was too difficult to do. I collected all the samples actually after I finished all of the rest of my PhD work, and didn't actually do the analyses until 10 years later when I was an assistant professor. And so, but it allowed me to, see lots of different places in Hawaii. Oh, I forgot I had animation. So it colonized all the different islands, and we did a really kind of fun population genetic study of the colonization of bush warblers across the islands. But I ended up doing my field work on the island of Maui, and I was really interested in competition between native and non-native birds. And so we have Hawaiian makihi that's there, and then we have Maui alauahio. And these are two of the main insectivorous birds that are there. And then in these forests, there are a range of different introduced birds. And these are three of the most common. And so Japanese bush warblers, Japanese white-eyes, and red-billed leothrics. So really wanted to know, are these birds competing over food resources? Are they eating the same arthropods that are out there? And so here's the part where I got into a lot of bird poop. And this was actually, I think, my grandmother had really identified this. She gave me this book, I think when I was 8 years old. It was a long time ago. And so she gave me this book, and it was like an omen, I think. And this is the more current version, How to Be a Poop Detective. And I've kind of continued kind of along this track. And so this is a red-billed leothrix in the net. And this is exactly what we're collecting from those birds. And this is just a cool aside. This is not a bird poop. This is a caterpillar that looks and is a bird poop mimic. Isn't that unbelievable? Totally, totally crazy. And a total aside, even though caterpillars are the major part of almost all of these bird diets. This is me in the early 2000s up in a tree when I used to do field work. This is before that branch broke and nearly skewered myself. And so here's what I did. Here's what I did for 2 years of my PhD. I went in and identified every insect or arthropod. There's lots of spiders as well. Every arthropod that was in these samples. Amazingly tedious and time consuming, but I got really good at being able to identify, sometimes to the species, what the insect parts were. So I identified over 4,000 invertebrates and seeds from 252 samples. And diets contain 18 invertebrate orders and 18 plant species. And this is a bit of an aside. It turns out, again, the project that I had planned was too difficult. But it was too difficult to really focus on the insects, at least the insect portion of it. And really a lot of interesting stuff was happening on the seed side. And it turns out nearly all these birds were eating native and non-native plant seeds. And so that took me off in a different direction and allowed me to collaborate with Don Drake and others. And so that was kind of a separate thing that we're not going to talk about today. And so we're going to focus on the insects, even though it was really difficult at that time to be able to do it. And I vowed to kind of do better. But kind of at a really broad level, we can see that the bird diets overlap. But if you look here, this is at the level of order. And so you can't identify every insect part to the actual species, at least not morphologically. And so at this level, this is a really, not an ideal way to compare kind of diets in that, there's lots of, this was Homoptera at the time. There's no entomologists here, are there? All right.
Paul Kershaw is on here.
All right. Oh, Paul. Yeah, I knew someone was gonna call me up for that. These have two merged since then. But so the main thing here is that at the order level, you can't really tell what's going on because there are lots of different insects in each of these orders. And to say that they're overlapping is certainly overstating the data. And so the results definitely left me wanting more. I did not want to do all the insect sorting or arthropod sorting. It took considerable effort. I had to, at the time, this was pre-email for a lot of people, particularly the entomologists. Most of them, even at that time, were not in the new ages, and they did everything by mail. And so I'd have to mail out kind of pictures and mail out specimens and hope for that they would respond to me. And it turns out most of them did. It was amazing kind of network. Limited ability for species level resolution. Most of the time we didn't know what species were in our samples. And so it was very difficult. And we needed just more robust sampling. And so we really know that these insect and arthropod communities kind of change temporally and spatially, but really it's difficult to do, especially in your dissertation, at a limited number of field sites and limited number of sampling periods. An additional kind of question is, are arthropods for birds really a limited resource? And so are they truly competing over this resource or not? So I vowed to not do molecular identifications with microscopy and really wanted to use some new method. But it turns out that method wasn't available until, well after this. And so two things have really changed since this time. Insects have been in major decline and globally, and the technology has changed. And so some added benefits. And so insect declines various insect groups are declining globally. And so some groups more so than others, but it's a common pattern throughout the world. And so this is just some images from an article in Science from 2014. We can see here Lepidoptera, so the moss and butterflies are declining, and all other invertebrates are in sharp decline as well. And it's particular groups or particular orders that have the major declines. And so beetles and ants and bees and wasps and the Lepidoptera and then the dragonflies and related within Odonata are all in pretty major declines. And so it turns out birds are in decline as well. And so in many cases, the same birds that are declining because of declining insect resources. It's not true in every case. For example, grassland birds are as affected by declining habitat quality and availability, but at the same time, they're insect prey, and I say insect and arthropods kind of interchangeably. But in this case, it's insects and other arthropods like spiders are in decline in many of these grasslands. And so it turns out we're working in all of these forests on fecal metabarcoding, except for Arctic tundra birds. I'm going to show you 3 examples today from some of these groups. So this is what you can see with a microscope. You saw those kind of focused images. And again, you get these kind of major groupings. Sometimes, yes, you can identify the species or genus, perhaps of an arthropod within a sample, but in general, you can't get that. And so you can see a lot more if you use genetics. And so if you use down to the molecule level. So you collect the fecal sample, you extract the DNA, you amplify that with PCR, so you make lots of different copies, you sequence that, and then you analyze it. So all pretty simple. Seemingly simple. So metabarcoding is easy, just 4 simple steps. The key thing here is that we have primers that selectively amplify a gene that is found in all animals. And so all animals have this particular gene, and if you amplify that gene, you can and then sequence it and then compare it to a database of sequences, we can determine what species that this gene came from. And so if it's a particular sequence here, we know it came from this particular species of beetle. We know this one came from a moth. We know this one came from a fly, and so on and so forth. And we know the exact species of each of those. One challenge, though, is that they're coming from animals. And so oftentimes, especially if the primers aren't great, we amplify a lot of the host that gave you the sample. And so, so this was, you can see that I have a bunch of different puns in here. This was actually one of my graduate students, Devin O'Rourke. It turns out building these databases of sequences that you're comparing them to and kind of running these analyses, you have all these assumptions kind of built into them. And it's actually quite difficult. And so this was a way to kind of formalize that and be kind of more restricted in exactly how you're gonna kind of collect your samples and analyze your samples and compare what you found to the database of millions, if not billions of sequences that are out there that have been identified. So the short version of that is that every step matters, and you really have to have an understanding of every single step. And the sequence processing can affect your inferences or your analyses, the way you build your database of sequences, known sequences that you're comparing it to, how you classify those sequences is all difficult. And I'll show you some examples from Hawaii at the end of where it continues to be difficult, especially here. And so the first study I'm going to talk about is from diets of the eastern whippoorwill. And it turns out Audubon already knew what they ate. And so we can see here that Audubon is depicting that eastern whippoorwills eat moss. And it turns out they ate moss in the 1800s and they eat moss now. So we already knew, so maybe we didn't need to do this. And so, but eastern whippoorwills, this is their breeding population here, kind of in this orangey pink here. And so we can see that percent declines are pretty big throughout much of the eastern United States. So this is a species that is an actually pretty sharp decline throughout much of its range. And so this is just an index of its population decline. And you can see here that based on this index that whippoorwills are indeed in sharp decline. So this was with collaborators Ian Souza Cole, who's now with Audubon in California. He was a grad student of Mike Ward and TJ Benson. And so they sampled at a bunch of different sites, sampling both whippoorwills and the moths that were present. And they focused on moths because they already knew that even before our data that moths were an important part of the whippoorwill diets. And so this is the state of Illinois. And so they sampled extensively in these areas. And no surprise that whippoorwills eat moss. And again, I'm kind of showing only at the order level, even though we have species level identifications for nearly all of these insects. And so again, you know, nearly 90% of the samples contained Lepidoptera and contained in lesser amounts flies, cockroaches, and beetles. It turns out nearly all the samples that we have from almost any habitat have cockroaches in them. Again, we're trying to figure this out, but certainly an abundant diet item for these whippoorwills. And lots of things to kind of talk about, but one thing that we can find, and so this is on the y-axis, this is whippoorwill abundance, We can see here, this is at the highest level and from the forest edge. And so the areas with the highest moth abundance here that we see here around 40 meters from the forest edge are the areas of the highest moth abundance. And so areas where moths are abundant, which coincides with areas that are distant from the forest edge, have the highest whippoorwill abundance. And so kind of on the basis of this, we built a grant proposal, not only to look at diets, but to also try to link them to habitat quality. And the punchline here is that we don't know yet, but we're kind of building towards that. And these are kind of some of the steps on how we're trying to identify what the birds are eating and from their fecal samples, doing the metabarcoding to identify which insects or arthropods that they're eating, and then connect them to different plant species that are there. And so this is a collaboration with a bunch of different groups. This is just one of the sites. And so this is at Fort Drum, New York, as well as additional sites in the Eastern US. And so Amber Roth is leading this effort in her lab. And two of the species that are in pretty sharp decline are kind of these grassland savanna species, golden-winged warblers and blue-winged warblers. It turns out they're closely related to each other. You can visibly see the differences between them, but they are in the same genus and they will hybridize. And so they can interbreed. And so no surprise, and you don't need to see all these details. Golden winged warblers eat mostly caterpillars. And so unlike what we saw in the whippoorwills, these are, the warblers are eating almost always the larval stage, not the adult stage. So the whippoorwills are eating adult moths, and the warblers are eating mostly the caterpillars. We can't actually tell that from this data. From these data, we don't have any idea whether they're adults or young. We just know exactly how they're feeding. But you can sort this better just based on the caterpillars, and you can see that at least half the samples have the majority of Lepidoptera in there. And so each one of these on the x-axis is an individual sample, and the height of the bar indicates the percentage that it is within those different groups. And you can see here this green group that they're all eating, almost all eating caterpillars, and some of them are eating almost entirely caterpillars. Again, we have species level resolution that I'm not going to talk about today, except for kind of one exception, again, for the entomologists. And so thanks to Paul for being on the call. Maybe some other entomologists are out there. And so it turns out if we look at golden winged and blue winged warblers, they eat almost exactly the same thing. And so even though these are different species, they are eating almost exactly the same diets. And so we can see here that, they're largely, for blue winged warblers, they're largely eating moss. Golden winged warblers, again, are largely eating moss. And then they're eating roughly the same amount of kind of other assorted arthropods out there. And if you have a close eye, you'll see that they're eating a lot of Hymenoptera, which was not too surprising since I've actually seen it a lot in the Hawaiian bird diet data. Anyone know why they're eating lots of Hymenoptera?
They're eating parasitoids?
Yes, great. Maybe I should ask everyone else, but yes, they're almost certainly eating parasitoids. These parasitoids parasitize the caterpillar, the larval stage of Lepidoptera. And so we know that because we know exactly which species they are. And so, and indeed, a bunch of the flies, there's several fly parasitoids as well that parasitize caterpillars as well. And so we're seeing kind of the secondary level of into kind of this food web beyond just exactly what the birds are eating. Again, kind of this is NMDS plot to kind of look at the differences or similarities in their diets. In an ideal plot, you would see the blue group here. Blue group, I totally missed the chance of having blue-winged warbler be blue. But normally you see kind of, if you see this clustering, blue-winged warbler diets would be up here. And then golden-winged warbler diets would be down here. And if they're hybrids, they would be somewhere in the middle. We do not see that at all in terms of their diets, kind of in this non-dimensional space. But we can see that they really overlap considerably. But we really wanted to know more beyond this, and our data aren't out there yet, but we're interested more than just what they're eating, but exactly the quality of their dietary items. And we know that these two biomolecules are directly related to growth and survival of nestling birds. And so we're looking at carotenoids and omega-3 fatty acids to try to understand if different habitats that these birds are foraging in are related to different measures of productivity. So we actually know how many nestlings fledge from a particular nest, what their survival is, juvenile survival. And so we're trying to link exactly the food items, but also what's contained within the food items in these to potentially understand what is good and poor quality habitat. Can we kind of make those connections? And so I'll have to come back in a couple of years with the results. But kind of an added aspect is that insect prey may also be declining in quality. And so another reason to kind of really look at these insects, some really interesting data kind of talking about this hypothesis of nutrient dilution. It's not too complicated, but the idea is that as with global warming, we've had reduced amounts of nutrients within these. Basically, the nutritional quality of the plants is getting diluted, and so the insects are losing their kind of quality, and that may be directly affecting bird populations. And so a total shift here, but just kind of another way that we can use this exact same primer set. So I told you that this primer set works for animals. And so there's lots of other things besides arthropods that are animals as well. And so we're looking at purple sandpiper diets in the Gulf of Maine. And so this is a collaboration with Elliot Johnston and Brian Olson and a bunch of other collaborators within the state of Maine. And so they're both at the University of Maine. And this was part of Elliot's dissertation. And so we can see here the non-breeding or wintering range here, and we can see the breeding range kind of within the near Arctic and Palearctic. And so we can see here that, they're in the winter, they're on the coast of Maine. But not surprisingly, they're in the shorebird group and they're in rapid decline. And so we're trying to see, is diet a part of that decline? And so in Maine, 49% decline from 2004 to 2014. And so this is just one relationship that we know about. We can see here on the x-axis date, and then we can see the month. The key thing here is that everything on the right-hand side, or nearly everything on the right-hand side, is in red. And so we have warming temperatures in most months throughout the Gulf of Maine. So the Gulf of Maine is really increasing in sea surface temperature and potential effects on the entire food web. So a bunch of different sampling sites in eastern Maine. And so here's what they had to do, and this was based on their kind of safety requirements, although they take these boats out to these rocky outcrops and go out and collect it. And certainly it's cumbersome to wear a Gumby suit when you're kind of on these areas, but the ocean is 35 degrees and it's the middle of winter. And so they have to take these precautions, going out and seeing where the purple sandpipers have defecated on the rocks, or catching the purple sandpipers and having them defecate within the chamber that they provide. So we're interested in what are the purple sandpipers eating, and then kind of getting an estimate of what they're eating based on the DNA that's in their fecal samples. Again, you've kind of heard about this. And so very similar thing that I'm going to show you. Each vertical bar is a diet of an individual purple sandpiper. And much like what you saw in the golden-winged warblers, is that they're eating almost entirely caterpillars. And that wasn't a surprise. They're eating almost entirely semi-balanus. And so this is a barnacle. And so they're eating a specific species of barnacle. And purple sandpipers do not eat barnacles. And so we were like, this is confusing. Something must be wrong. And so we submitted this to a journal. We're like, there's nothing wrong with this. The reviewers were like, something's wrong. You need to do all these other tests. And so we did all these other tests. It took like 9 months. And it turns out they eat mostly barnacles. And so we're seeing a major shift in their diet from what is traditionally their diets of blue mussels, and we'll see that in subsequent slides, to most sites they're eating mostly barnacles. And barnacles are believed to be lower nutritional quality. They're certainly a smaller diet item, so they're going to have to eat a lot more barnacles to get the equivalent nutrition of a single blue mussel, for example. And so we're seeing major shifts that we believe are related to warming temperatures, and Elliot and others are kind of still looking into that. So it appears, at least from initial glance, that the diet shifts may be leading to these population declines. Warming temperatures are leading to differential prey in the environment, the loss of blue mussels, and the increasing dependence of purple sandpipers feeding on barnacles. And so we've had lots of successes, but many challenges as well with fecal barcoding. And so really the idea is kind of getting beyond these descriptive studies. And so one thing that we're looking at is, can we use these as a measure of habitat quality? Additionally, it's really, with declining insect population worldwide, it's kind of really difficult to understand because it's kind of this moving target. Also, I found out from my dissertation and kind of continued to find out, assessing competition is hard. And so if any of you want to look at competition, I'd suggest you stay away from it, even though we have really good tools now to really understand at the species level exactly what they're eating. So maybe you can do it now. But I think a major challenge is that knowing if arthropods are limiting in the environment is really difficult. But we do know that, especially from the whippoorwill study, that we can actually measure the abundance of different insects in the environment, like moths. We can use this to determine where on the landscape animals are foraging. And so I didn't show these data, but oftentimes the insects in our studies have specific host plants, and we know exactly which plant that they got that insect from, because that insect is always found on a particular plant host. And so we're going to kind of continue to use that to understand, are there particular relationships between where birds are foraging and on particular plants. Again, I kind of didn't show this today, but we have lots of data from both birds and bats where we can be the first line of detection. And so we can use the animal diets as an indicator of when new invasive pests have moved into a site. And so I'll never forget, I talked to the state forester in New Hampshire when we were working in forestry, and I said, we just found this new sawfly here. This is an invasive species. And he was like, how'd you know? We just found this last week. And so we simultaneously detected it through two very different ways, but it's an amazingly sensitive way to kind of pick out particular insects or arthropods out of the environment. And so much of what was developed was for microbes, and so microbiome work, and there's a whole institute that does microbiome work. And so much of the methods were kind of developed for that, and they're really advanced for that. And so it's all about kind of using those same tools to look at it for these marker genes for other things like CO1 for insects in animal diets. And so it's a really good template, but we have to kind of modify it slightly. And there's lots of kind of buts here. And so as Paul will kind of attest to with the challenges that we're having with his data sets. But reference data sets are incomplete, and I'll show you on the next slide kind of exactly why that is in Hawaii. Contamination is always out there. Species level, I said we can usually get species level, yes, except when it really matters. And in those cases, we can't. And the two different species have the identical barcodes. Really, really challenging. Usually that doesn't happen, but sometimes it does happen. Misidentification or not detecting taxa that we know are present certainly happens. Difficult to quantify. We don't know how many of a particular insect are in a sample. Usually, we just know roughly how many reads are in a sample or how many sequences, but oftentimes that is not closely correlated with the amount or the importance of that species in a diet. And we have only correlational data linking bird diets to human-induced changes. Yes, it's a great story to kind of link purple sandpiper diets to increasing water temperatures, but we don't know the exact mechanism yet there. And so what are we doing now? And so we're like the microbial people that are sequencing the world. We're sequencing all the poop that we can get. And so we have, I didn't show this today. I had a graduate student, Sarah Gabrielson, who, did she present to you guys? Maybe to somebody else. And so at UH. And so we have diet data from rats, black rats, and mice, house mice in Hawaii and other rat species. We know exactly what they're eating in Hawaii. I didn't show you those data here. We've collected over 4,000 samples of native and non-native birds throughout the Hawaiian Islands and are looking at their diets there. We have game bird diets. We're about to start barn owl diets. And so lots of metabarcoding. All right, so I'll ask you one additional question. So if you go into a native forest and you're looking at insectivorous bird diets, and so these are high elevation native forests, what is the #1 plant that you find within the fecal samples? Don can't say.
Moss.
Moss. All right, you say moss,
That's
I
my
like
guess,
that.
yeah.
Oh, yeah,
Oh, yeah. Why'd you say that?
because native, I see a lot of native birds on ocean.
Yes, but in this case, we're getting the secondary level. It's almost always in the insects that the birds are eating and most of these aren't actually consuming ohia directly. And so we can see that kind of second level of the food web in terms of nearly all these insects are eating ohia, and especially in these forests. And when we go to other forests, we don't see it. We see them eating other plants there, but we certainly see that strong signal of ohia, even though many of these birds aren't consuming ohia directly. And so to quickly get to Hawaii, and so as an entomologist once told me, insects are animals too, and I said, but real animals, and he got offended. But insects are animals too, right? But only 23.8% of the arthropod species in Hawaii have barcodes. So we only have barcodes from less than 2,500 of the over 10,000 taxa that we have here. And this is a figure generated by Dimitrios Patsopoulos as part of a project that George Roderick and Rosie Gillespie from Berkeley, Natalie Graham from Hilo, Paul Kay from here, Mark Johnson from the Army, and many others from across Hawaii and the Pacific. And continuing and as well as a new project funded by the military to kind of look at arthropods throughout the Pacific. The key thing here is that in pink, we can see what's been sequenced, and what's in gray is what's not sequenced in each of the orders. And so we can see here, you know, some groups are really well done, but it's only because they have a handful of species in the entire order. But for the most part, we've sequenced only a fraction. And so if we get a particular cricket from a forest in Maui, the likelihood is that we're not going to be able to identify the exact species. And so we're really kind of pouring our efforts into sequencing more insects and other arthropods from throughout Hawaii. And kind of continuing with Dimitrios's kind of schematic, it turns out this is very, as I tried to allude to in the total crapshoot slide, turns out this is very difficult. The same species may have different names. The genus may have changed. The species may have changed. So resolve synonyms. And so 10,295 species with a valid status, but many of them have the same names or conflicting names, or they change over time. There's different databases that you're pulling them from. And so those different databases curate the data differently. You have to create a master list, merge the names, find the sequences. We don't all use the same exact region of that gene. So we have to make sure our gene is there. And so there's lots and lots of challenges. And at the end of this, only less than a quarter have been identified anyways. So most of the time we have no idea what species that we have. And so kind of I'll end on where we're going with this. And so this is a really amazing data set that shows from African savannas, really showing the relationship between a bunch of grazers and what plants that they are feeding on. And so most of these are grasses and forbs that they're kind of feeding on from the ground. And so we can see here, kind of these amazing connections and knowing exactly kind of what the buffalo are eating in various groups. And so this is a relatively, all the amazing data set, but this is just a bipartite network. And so there's just comparisons of two. And so what we have here, and you don't need to, I intentionally didn't allow you to kind of understand what's seeing here. This is a tripartite network. And so we have, one group here. It turns out these are parasitoids of the second group. And then the second group is all feeding on these different plants. And so we have kind of 3 levels here, but it turns out in Hawaii, even though the Hawaiian system is pretty simplified, we have far more than that. And so I can't see exactly what I wrote, but I wrote it out. And so we have the birds that are eating the insects that are eating the plants, right? And so we saw that. So that's a simple network that we have here. But we also, in Hawaii, we have the birds that are eating the caterpillars, but the caterpillars have the parasitoids in them. It's true in golden winged warbler diets, and it's just as true here. We have the birds that are eating the spiders that are eating the insects that are eating the plants. And sometimes we can see the insects within the spiders. And so we know exactly what the spiders are eating based on what's found within the bird diets, which creates obvious complications, right? We can see that the rodents, the rats, are eating the birds. And the rodents are also eating the insects, they're also eating the spiders, they're also eating the snails. And so we can see that rapidly kind of getting much and more difficult in terms of the complexities of this network. So again, I'm trying to wrap my mind around exactly how these networks are really interacting with each other, but we at least have a start with a bunch of these different bird and mammal species. With that, I have to thank a lot of people. And so this is Devin O'Rourke and Becky Mao, who did a lot of the analyses, as well as that was in the past, as well as Katie Priest, John Galeese, and Alexandra Gibson, as well as the kind of heavy hitters on the bioinformatics part, as well as the collaborators I talked about in those other studies. And with that, I will end and take questions. Thanks.